Hello and welcome to the third series of Enzymology for Recombinant DNA Technology series. Myself, your host, Dr. Deepak Verma. Today we will discuss about restriction from the restriction endonuclease. In first series we have discussed about ligase, and in the second series we discussed about nuclease. Now, in this series, we will discuss about restriction endonuclease. That these enzymes are very important enzymes for this biotechnology field. Restriction endonucleases are a group of DNAs that recognize a specific nucleotide sequence and cut double-strand DNA in a site-specific or non-specific manner. They were initially discovered in the early 1950s as a part of the restriction and modification system that bacteria operate for their protection against invading bacteriophage and foreign genetic elements. Initially identified by phenotype, the restriction modification system are now changed to a wealth of genetic analysis. It is largely through this restriction modification system that bacteria destroy undesirable foreign DNA that invade the cells through infection, conjugation, and transfection. The bacterial restriction modification systems are tantamount for the prokaryotic equivalent of an immune system. This restriction modification system generally consists of two enzymatic activities. Number one is site-specific restriction endonuclease that is responsible for digesting exogenous DNA, and next one is the DNA modification that is also known as methylases or methyltransferase with identical sequence specificity. There are there are four class of restriction endonuclease, but most of the literature because showing this only three types: type 1, type 2, type 3. So restriction endonuclease type 1 enzymes are multi-enzyme complexes composed of three non-identical subunits. Restriction number 1 is restriction, number 2 is modification, and number 3 is S. The S subunit determines the specificity for both R and M. The type 1 restriction modification enzyme requires magnesium ion and ATP as a cofactor and cleave unmodified DNA at the relatively random location distant from the recognition site. Type 1 enzyme hydrolyzes ATP as part of the restriction activity and site-specifically methylated the target DNA in a manner competitive with cognate DNA activity. Now comes to the type 2. This system consists of physically separate RNAs and methyltransferase activities. These restrictions are usually dimers having identical subunits, whereas methyltransferase are monomeric enzymes. Type 2 restriction endonucleases requires only magnesium ion for their restriction activity. Type 2 restriction endonuclease and methyltransferase recognize the same specific nucleotide sequences. The majority of type 2 restriction endonuclease cleaves DNA at the recognition sequence, while a small number of enzymes called type 2S cleave the DNA at defined distance from the recognition site. Now we comes to the last. Type 3 restriction modification enzymes are composed of two non-identical subunits and require magnesium ion and ATP for restriction activity. Type 3 enzymes recognize short non-palindromic sequences and cut DNA at fixed sites relatively to unmodified recognition sequence. Type 3 enzymes do not hydrolyze ATP. Adomet is not required for restriction, but it stimulates the endonuclease activity. Now we come to the type 1 in details. Type 1 enzymes are probably the most complex of restriction modification system. They are made up of 3 subunits, requires 2 cofactors and have at least 3 enzyme activities. Several type 1 restriction modification systems have been identified in Escherichia coli, Salmonella and Citrobacter. Type 1 enzymes fall into the 3 distinct families depending on their gene organization. The chromosomally encoded K and A family and plasmid encoded family. The type 1 restriction enzymes are encoded by HSD gene, host specificity for DNA. Among the type 1 enzyme, EcoK1 and EcoB1 are the most extensively characterized in terms of genetic structure and function. Based on the 2K family enzymes, which have proven themselves useful in recombinant DNA technology, here we describe some specificity for these 2 enzymes. The international unit definition is written here. And now we come to the direct specificity. First is the recognition sequence. As DNA binding and cleavage by type 1 enzyme depends on the presence of host specificity site, both EcoB1 and EcoK1, and in fact all type 1 restriction modification enzyme recognize specific bipartite sequence. Means denoted by the asterisk indicates that methylation site. The general structure consists of a specific heptameric sequence divided into a trimer and a tetramer by non-specific spacer sequence of fixed but different length for the 2 enzymes. Now we come to the another one, DNA cleavage. Endonuclease action occurs only on unmodified double-strand DNA. The DNA cleavage takes place away from the recognition site more than 400 base pair away to as far as 700 base pair. The process of DNA cleavage at distinct location on either side of the specificity site most likely involves a loop-mediated translocation of the DNA. The pattern of cleavage is not totally random, but is localized in a part probably related to the structural feature of the DNA. Now the last one, DNA methylation. For both EcoB1 and EcoK1 system, the modification occurs at A base. On both strands, 2 methyl groups are incorporated per site. For EcoB1, the top strand contains a single A, and this must be the one that is methylated. The lower strand contains 3 adenines. The middle A is the one that is methylated. For EcoK1, the most likely site of the methylation are the single A in the lower strand and the second A is the top strand. Now a application, these 2 enzymes have some sort of application, but it is not useful until now. Come to the type of site specific R1 restriction of modification enzymes are probably the best of all type 2 restriction modification systems in terms of structure and function. Restriction EcoR1 specifically recognizes a palindromic hexanucleotide sequence 5' GAATTC' in double-strand DNA and cuts the phosphodiester bond between G and E, producing a 5' protruding 4-base terminal. EcoR1 is the first restriction endonuclease to be purified to homogeneity and in a large quantity. EcoR1 endonuclease serves as the prototype of ribonucleases in detailed understanding of their immunonucleation and DNA interaction. Here, international unity is clear to the substance. Specifically, first is the recognition sequence. As EcoR1 recognizes the hexanucleotide characteristic sequence of 5' G AATTC in double-strand DNA and fills the phosphodiester bonds on both strands between G and in base. Now comes to the chain length dependence. Chain length this enzyme cuts the restriction site with a preference for either a smaller or large size of polynucleotide depending on the ionic strength of the medium. At O-ionic strength, the cleavage rate on PBR322 is 12 times higher than on the 34 base fragment derived from PBR322 and containing the EcoR1 site in this central location. That similar ionic strength, cleavage occurs in least partially in processing mode by several hundred years. Now the site preference and polarity. All canonical sites in given polynucleotides are not cut equally. With for lambda DNA, the site nearest the right F terminus approximately 10 times as far as site in the middle of the molecules. This apparent polarity is also shown in several other restriction enzymes and in varying degree depends on the substrate DNA. We come to the effect of methylation and desubstitution. When either one hemimethylation or full methylation of central A are methylated by EcoR1 methylase, EcoR1 can no longer cleave the D. EcoR1 is also sensitive to methylation at non-canonical sites and does not cleave methylated guanosine guanosine site. EcoR1 source is associated to cleavage as hemimethylated sequences. Now, modulation of site is like other type 2 restriction enzyme. EcoR1 is not only sequence specific but also structural specific. Some DNA interfactor and ligands binding EcoR1 reversibly modifies the site presence of EcoR1 and results in varying degree of inhibition on EcoR1 activity. Now, DNA structural dependence. We know that DNA structure is also very important for as ZDNA, in case of ZDNA, the contrast to the usual recognition sequence DNA, the left and the ZDNA confirmation within the recognition sequence or at the distance of 4 or less is indicated by the EcoR1. And sometimes using the DNA triplex form. So, when the formation of a triplex helix acidic filament, approximately pH of 6.5, then the flanking region, the DNA, the flanking region of DNA, the EcoR1 recognized sequence inhibits the EcoR1 activity at a particular site without affecting the other site. Such form oligonucleotide directed inhibition of DNA cleavage is site specific and reversible, and some other structures also having some role in sensitivity, such as can say that the endonucleatic activity on double strand DNA is same regardless of its conformation, whether supercoiled This is cDNA, for 1 on open circular DNA, for 2 on linear molecules. Now we come to the functions of this active registration agent. This actually these functions are very short. So okay, we will start. Active restriction in nucleotide. As we know, there is a large group of DNA nucleotides that is used from the restriction nucleotide system like functionally separated from the uptake modification based on clearance and nucleotide sequence specificities of restriction and enzymes are critical type using the restriction in nucleotide type specific DNA sequence and within the molecule. Here we start from specificity in type 2 restriction enzyme recognize specific sequence along the genes and cut them into the recognition site or at the defined number of nucleotide systems on the recognition site. The majority of type 2 restriction nucleases recognize tetra and hexanucleotides. There are barely over 10 in base cutters known. Known by a vertical bar. Okay. Another terminal structure of cleavage. The cleavage involves the isolation of two phosphodiester bond open for extent either sequentially or simultaneously. The 5' phosphate and 3' hydroxyl terminal can be either blunt or flash and or 5' or 3' representing offensive or sticky edge with various numbers to the 6-base extension. Now, site structures are the recognition sequence of the majority of type restriction in the nucleotide system. Inverted complementary sequence are palindromic. The cleavage occurs in the recognition sequence. Since the DNA confirmation restriction endonuclease are not only sequence specific but also structure sensitive and may fit either enhanced or unique cleavage activity. The restriction site located at nuclease S1 sensitive confirmationally flexible section. For example, between formation pDNA and ZDNA or in non-pDNA confirmation are resistant to cleavage by the many restriction enzymes such as EcoR1, PIN3, PST1 and DOT. Next is the DNA-RNA peptidonuclease. Several restriction enzymes are able to cleave DNA-RNA hybrids, albeit at slower rate or 20-50 times higher enzyme concentration. These enzymes include EcoR1, PIN3, HHK1, PIN3, MFP1, and Z1, PIN3. Now we come to the isosomers. Often, with the given canonical DNA sequence is recognized and cleaved by more than one enzyme. Such group of enzymes are generally referred as isosomers. Isosomers recognize identical sequences, but the restriction pattern may not be the same. More specifically, those enzymes that cleave at a position differ from prototypes of neosomers. Regenerate recognition, next is the regenerate recognition sequence. Some restrictions in nucleotides accommodate more than one base at a particular position in sequences and cleave them with equal efficiency. This regenerating can occur at any one of the two base positions with a current base sequence in such a way with some symmetry about the inversion or any rate distribution is maintained. Next is the relaxation of site specificity. Type 2 restriction enzymes generally type rigorous sequence discrimination by a factor of greater than 10 to the power 6 between the canonical site and other alternative reverse sequences. However, other suboptimal conditions, a significant number of enzymes display moderate cleavage activities on slightly different recognition sequence as well as recognition sequence containing a single base nucleotide. Now the increase of site specificity. Type 2 restriction endonuclease maintain the capability of specific sequence recognition and cleavage, although the rigor of the site specificity can be relaxed under certain reaction conditions. Indeed, site specificity can be increased under some of other reaction conditions by applying combinations of strategies and by modulating flanking sequences into sequence composed of higher number of nucleotides by means of the methylation. Now the reversible modulation of restriction enzymes. The site specificity of restriction enzyme can be reversibly modulated by DNA binding ligand, interaction, DNA binding proteins, or triplex forming oligonucleotides. Next is the restriction site containing base analog. Reversible site modifications such as both base modification and substitution can seriously affect the action of certain restriction enzymes. Last is the monitoring reaction activity. Sequence specificity of restriction enzymes can be differentiated from one another by gel analysis of their characteristic cleavage pattern on some strands DNA such as SU40, 5X174, plasmidemia 3. Sequence specificity as well as the activity of some restriction enzymes is strongly dependent on reaction conditions such as the quantity of DNA, ionic strength, that is salt concentration, pH, and temperature. Fortunately, some enzymes have similar optimal reaction conditions which allow for the grouping into the several restriction buffer systems. Indeed, such a grouping is not only convenient or delayed with a wide variety of enzymes, but is a way necessary to maintain consistency in data comparison among enzyme users. Now we come to the application of type 2 restriction enzyme. First is the universal restriction. The separation between the recognition site and the cleavage site has been exploited to make the universal restriction enzyme for cutting DNA any target site. The recognition specificity is provided by the self-complementary hairpin part of the oligonucleotide adapter, which the target is supported by a single strand primer part that would be complementary to the target sequence. The gene synthesis. This method of gene synthesis is based on the unique property of the to generate 4 ways including terminal, which can be used for ordered ligation of oligonucleotide. The synthetic oligonucleotide is first released between the two edging sites in a hedge image of plasmid DNA, then transformation of the DNA loop in vivo gap repair, and bridge mutation. Next is the agricultural approach of gene cloning. DNA cloning often involves insertion of the DNA structure into the cloning factor by a intramolecular ligation compatible with it. In some cases, type 2 restriction can recognize different sequences yet produce identical protein ends and facilitate a cloning task. More often, however, commonly used restriction enzymes generate proteins and that are not compatible with those generated by the other enzymes. When the gene fragment includes contain internal restriction site, the situation require either the consideration of alternatively insertion strategies or misuse of more complicated steps such as protection of internal site by methylation. And as a also one of the applications as a versatile tool for gene screening. This technique implies the plasmid in which the unique nuclease recognition site is present in or flanking the multiple cloning sites. A DNA fragment cloned into the multiple cloning site can be progressively trimmed from one end. Larger deletion can be performed by multiple cycles, cleavage, and move being nuclease digestion and religation. Next is the oligonucleotide amplification ratio. When the protruding nucleotides cohesive and generated by endonucleotidization is filled with using polymerase, the terminal can be recruited by the restriction enzyme because of the recognition site remain intact. In the presence of both polymerase and restriction endonuclease cycle continues, generating copies of amount of tetramer which is complementary to the cohesive nucleotide end. Now locating methylated base in the DNA. This method is based on the observation that the type 2S restriction enzymes are not inhibited by the methylation at a cleavage site. Briefly, a DNA fragment is chosen such that it contains the recognition sites of the methyl transferases under study and that the site appropriately overlaps a type 2S enzyme cleavage site. Last is the linker mutagenesis and the DNA replacement. In a type 2 restriction endonuclease which contains a selectively marker and is amplifiable separately in a specialized vector is placed in a linker area of gene closed in another specialized vector. By restriction digestion, the cassette is released together with the several nucleotides beyond the cassette vector fusion base. Cohesive and ligation of such deleted double-stranded oligonucleotide by RE adapters allow the introduction of defined mutagenesis sequences into the target gene. Now we come to our last topic, type 3 restriction endonuclease. These systems has so far only 4 known members. One encoded by E. coli, EcoP1, the second encoded by the resident plasmid P15B, that is known as EcoP15I, and the third and fourth apparently coded by the chromosomal of Haemophilus influenzae RF HIMS3 and STYLT1. Type 3 enzymes recognize short non-palindromic sequences. Type 3 enzymes require magnesium and ATP as a cofactor, but use them differently from the type 1 RM enzyme. Unlikely the type 1 enzyme, type 3 RM enzymes are composed of 2 non-identical subunits. They methylated only a single strand of recognition sequence and cleave DNA at defined site 3' to the unmodified recognition sequences. The important thing is here, this activity is stimulated by the addition of adomet and substrate specificity. These are very recognition sequence. There are only 4, so there are only not restrict enzyme. And then we come to the DNA cleavage. Type 3 enzymes cleave DNA at about 25 to 27 base pair 3' to unmodified recognition sequence and generate fragments with 2 to 4 bases 5' protruding termini. The cleavage requires two unmodified recognition sites that can be separated by different distance but which must be in inverse orientation. This actually explains why some sites are not restricted and why complete digestion is impossible even in the absence of adornment. The last is the influence of flanking sequence. Type 3 RM enzymes do not cleave or methylate at each of these recognition sites with the same efficiency. EcoP1 and HIMS3 showed some dependence on the structure of the substrate DNA. Linear molecules of called E1 DNA are poorer substrate for HIMS3 than the supercoiled molecules. Lambda DNA fragments with the molecular weight of less than 2 to power 10 power 6 are not cleaved by the HIMS3 enzyme, but they can be methylated. And for application, type 3 restriction enzyme so far is not useful in recombinant DNA technology. So, as up to here, we have seen type 1, type 2, and type 3 enzymes, and we saw that type 2 enzymes are actually important and useful for recombinant DNA technology. The restriction endonuclease is a bacterial enzyme that cuts DNA into the fragments after recognizing specific nucleotide sequence known as recognition site. And up to 1,500 type 2 restriction enzymes, type 2 restriction endonuclease enzymes isolated from prokaryotic organism. And due to the isosuisovers, they recognize more than 130 nucleotide sequences, different, unique one, and they can scan a DNA molecule and stop. They are very specific for their substrate is very specific, so they scan the DNA molecule and stop where they recognize their sequences and control. They recognize by their sequences by the palindromic sequences. Here I want to tell you about what is the palindromic sequence. So palindromic sequence is the DNA sequence. This is a mirror sequence actually. See here T, T, A, G, C, A, C. Okay. So if we mirror these sequences, C, A, C, G, A, T, E. This is the actual mirror. See, this is the one strand and this is the another strand of DNA. So to avoid any type of confusion, the restriction enzyme class nomenclature is the following. Next slide. So the name of any restriction endonuclease consists of three parts. Number one is an abbreviation of genus and species of an organism of three letters, ECO. For example, ECO for Escherichia coli identified by the first letter of the genus and the first two letters of the species. The number two is a letter number or combination of two or indicate the strain of the relevant species. And the third is a Roman numerical to indicate the order in which different restriction modification systems are found in the same organism or state. For example, here I am going to only this EcoArval. The name of EcoArval restriction enzyme was derived as E stands for Escherichia, that is genus. CO stands for the coli, this is species. R is a RY13 strain, and I is the first identified order of identification in the bacteria. Here, this is a restriction site. This is showing that staggered and cohesive, and blunt end. So rather than cutting the DNA indiscriminately, a restriction enzyme cuts only double helical segment to contain a particular nucleotide sequence of 4 to 8 bases in length, known as restriction or recognition site. They are generally palindromic sequence. The position at which the restriction enzyme cuts is usually shown by the symbol slash or arrow. Restriction enzyme make either blunt or staggered cut. Thus, restriction fragments may have blunt end. The cleavage point occurs exactly at the axis of symmetry. Okay, this is the axis of symmetry. This one. So this type of product we have to find, and for overhanging and staggered cut, the cleavage point do not fall into symmetry axis, so the resulting restriction fragment crosses sticky or cohesive. So this cuts here and here and here. This type of cut. So this is the first product and this is the second product. And these are actually this is useful for mostly useful for this recombinant DNA technology. So these are some example enzyme names, and this table actually showing the enzyme name, source, and sequence, and what type of the product. So then BG code, all the things. And these are actually some important enzymes. These are all type 2 restriction enzymes. So thanks for listening up to here. The restriction enzyme are finished. Next we start with the alkaline phosphatase in the next video. Okay, thanks for listening. Okay, bye.